proteins have their own unique shape and purpose, or as many in the scientific community like to call it, their own form and function. To carry out their specific purpose, though, proteins must be in specific parts of the cell, or transported out of the cell. So, how do proteins get to where they need to be? Now that we've discussed how proteins are made in a cell, let's now move on to discussing how molecular labels are used to direct our newly formed proteins to different parts of the cell. Different proteins need to be sent to different parts of a eukaryotic cell, or in some cases, exported out of the cell and into the extracellular space. But how do the right proteins get to the right places? Cells have various shipping systems, kind of like a molecular version of the Postal Service, to make sure that proteins arrive at their correct destinations. In these systems, molecular labels, often amino acid sequences, are used to address proteins for delivery to specific locations. Let's take a look at how these shipping systems work. Translation of all proteins in a eukaryotic cell begins in the cytosol, except for a select few proteins that are made in mitochondria and chloroplasts. But that aside, translation of most proteins in eukaryotic cells begins in the cytosol. And as a protein is made, it passes step by step through a shipping quote-unquote decision tree. At each stage, the protein is checked for molecular tags to see if it needs to be rerouted to a different pathway or destination. The first major branch in this decision tree comes shortly after translation starts. At this point, the protein will either remain in the cytosol for the rest of translation or be fed into the endoplasmic reticulum as it is translated. If a protein that is still being formed through translation is transported to an organelle, again during translation, that transporting process is called co-translational transport. Co meaning with, so co-translational transport means transport happening with or at the same time as translation. Proteins are fed into the endoplasmic reticulum during translation if they have an amino acid sequence called a signal peptide, indicating that this should happen. In general, proteins bound for organelles in the endomembrane system or bound for the exterior of the cell must enter the endoplasmic reticulum at this stage. Proteins that do not have a signal peptide stay in the cytosol for the rest of translation. If they lack other address labels, they'll also stay in the cytosol permanently. However, if they have the right labels, they can be sent to the mitochondria, chloroplasts, peroxisomes, or nucleus after translation. Proteins destined for any part of the endomembrane system or the outside of the cell are brought to the endoplasmic reticulum during translation and fed in as they are made. This brings us to our discussion on signal peptides. The signal peptide that sends a protein into the endoplasmic reticulum during translation is a series of hydrophobic, or water-fearing, amino acids. These are usually found near the beginning of the protein. When this sequence sticks out of the ribosome, it's recognized by a protein complex called the signal recognition particle, or SRP. The signal recognition particle takes the ribosome to the endoplasmic reticulum. There, the ribosome feeds its amino acid chain into the endoplasmic reticulum lumen, or interior, as it is made. So, during translation, the signal recognition particle, shown here in orange, binds to the signal peptide as it emerges from the ribosome. The signal recognition particles then bring the ribosome to the endoplasmic reticulum by binding to a receptor on the surface of the endoplasmic reticulum. The receptor is associated with other proteins that make a pore, or a small hole. The ribosome resumes translation, feeding the polypeptide through this pore and into the interior of the endoplasmic reticulum. This interior we call the endoplasmic reticulum lumen. An enzyme called signal peptidase associated with this pore snips or cuts off the signal peptide. Translation continues, and the growing amino acid chain slides into the endoplasmic reticulum lumen. The completed polypeptide is released into the lumen where it floats freely. As shown in this example, in some cases the signal peptide is snipped off during translation and the finished protein is released into the interior of the endoplasmic reticulum. In other cases, the signal peptide or another stretch of hydrophobic amino acids gets embedded in the endoplasmic reticulum membrane. This creates a transmembrane, meaning membrane crossing, segment that anchors the protein to the membrane. In the endoplasmic reticulum, proteins fold into their correct shapes, and they also get sugar groups attached to them. Most proteins are then transported to the Golgi apparatus in membrane vesicles. Vesicles, remember, are super important because not only do they help different molecules get from point A to point B and to where they need to be, but they protect what's inside of them, right? Proteins, remember, need to be in very specific environments so they are not denatured. Vesicles ensure that the proteins get to where they need to be and that they remain in an environment that is conducive for the protein. So, most proteins are transported to the Golgi apparatus in membrane vesicles, but some proteins, however, need to stay in the endoplasmic reticulum and do their jobs there. These proteins have special amino acid tags, and these tags ensure they are shipped back to the endoplasmic reticulum if they happen to escape into the Golgi. In the Golgi apparatus, though, proteins may undergo more modifications, such as additions of sugar groups, before going on to their final destinations. These destinations include lysosomes, the plasma membrane, and the cell exterior. Some proteins need to do their jobs in the Golgi, and a variety of molecular signals, including amino acid tags and structural features, are used to keep them there or bring them back if they happen to leave. If they don't have any specific tags, proteins are sent from the Golgi to the cell surface, where they're secreted to the cell exterior if they are free-floating, or delivered to the plasma membrane if they are membrane embedded. This default pathway is shown in the diagram on the screen for a membrane protein colored in green that bears sugar groups colored in purple. Proteins are shipped to other destinations if they contain the right molecular labels. For example, proteins destined for the lysosome have a molecular tag consisting of a sugar with a phosphate group attached. In the Golgi apparatus, proteins with this tag are sorted into vesicles bound for the lysosome. Proteins that are made in the cytosol that do not enter the endoplasmic reticulum during translation may stay permanently in the cytosol. However, they may also be shipped to other non-membrane destinations in the cell. For instance, proteins bound for the mitochondria, chloroplasts, and peroxisomes are usually made in the cytosol and delivered after translation is complete. This is true for proteins bound for the nucleus as well. Because these proteins are delivered to their target organelle after translation in the cytosol is complete, the process of transporting these proteins is called post-translational transport. Post meaning after, so transport after translation. To be delivered to one of these organelles after translation, a protein must contain a specific amino acid address label. This label is recognized by other proteins in the cell, which help transport the protein to the right destination. As an example, let's consider delivery to the peroxisome, an organelle involved in detoxification. Proteins needed in the peroxisome have a specific sequence of amino acids called a peroxisomal targeting sequence, or a peroxisomal targeting signal. The classic signal consists of just 3 amino acids, serine, lysine, and leucine, found at the very end, or the C-terminus, of a protein. This pattern of amino acids is recognized by a helper protein in the cytosol, which brings the protein to the peroxisome. Mitochondrial, chloroplast, and nuclear targeting are generally similar to peroxisomal targeting. That is, a certain amino acid sequence sends the protein to its target organelle or a compartment inside that organelle. However, the nature of these address labels is different in each case. So, as a recap, proteins get to where they need to be in the cell either by being transported to the endoplasmic reticulum while translation is taking place, or by being transported to their target organelle after translation is complete. The first option is called co-translational transport, while the second is called post-translational transport. Proteins that are intended for the plasma membrane, lysosomes, endoplasmic reticulum, Golgi apparatus, or outside of the cell undergo co-translational transport. Proteins destined for the cytosol, nucleus, peroxisomes, mitochondria, and chloroplasts are transported to those locations after translation of the protein is complete, aka post-translational transport. As discussed before, all proteins have their own unique form and function. To carry out their specific functions, proteins must be in specific parts of the cell or transported out of the cell. We also already discussed how proteins are made in a cell, and now we know how molecular labels are used to direct our newly formed proteins to different parts of the cell. Awesome work!